Alright, here we are again with our sequence. It's the last gene in our contig, and I'm going to show you how to kind of find your transcription start site, given that we already know that our genes are near a CPG island. So we're going to start. I'm going to go set up as we have before in these examples, the location of our CGs and our double CGs. Alright, and so in this particular gene at the end, we've figured out that this is our CPG island, and here's our regulatory region above it. But what we're looking for here now is our transcription start site. And there are some ways, I'm going to show you some ways that you can find that, but they're not readily available to you guys. There's some software here I have that you don't have access to help you find that CPG island. But we're going to use the fact that we're in a CPG island to kind of find our transcription start site kind of in reverse, and I'll explain that. One of the promoters, you of course know about TATA boxes, and TATA boxes are usually not in CPG islands. Instead, the most common type of promoter in a CPG island is called the GC box. And I've checked these, and you should find GC boxes in every one of these. Now, if you know where the GC box is, you know pretty much where the transcription, or you potentially, let me rephrase it, you potentially know where the transcription start site is. And so that's what we're going to do. We're going to find our GC box first. It's like we're going to find our TATA first, and then from the TATA, we know about where it should be. You know, you can figure out where the polymerase started copying. That's the transcription start site. Well, in GC boxes, the GC boxes are quite a bit away. They're over 100 bases away from where transcription start site is. But we do have a pretty good idea of what the sequence is for the GC box, and I'm going to put this out, and I'm going to tell you to search first for GGG-CGG. That is the likely promoter sequence instead of TATA. This will be the sequence that more or less attracts your RNA polymerase. All right, and just it's a different sequence than TATA. There are other promoters, and this is a GC promoter, and they're the most typical ones found in CPG islands. And since all of ours that you guys have are CPG islands, this is a good sequence to use. It's probably the promoter that you're using, and so there's a really good chance of it. So we're going to highlight this one in green and see what we come up with. And we found exactly one. And guess where it is? It's right here. So this seems to have worked. Let's see, the absolute consensus are actually 4 Gs, a C, and 4 Gs, and sure enough, the general one, GGG-CGG, that I searched for was found, but actually the optimal sequence is actually 4 Gs, a C, and 4 more Gs, and that's exactly what's right here. This absolutely is a GC box. Now, the question is, if this is your GC box, where is your transcription start site? Well, there are probably many transcription start sites here, but I can tell you that in eukaryotes, now this is in a turtle though, but in eukaryotes it'd probably be a C followed by an A, somewhere much further down from your GC box. And if this were humans, it would be at exactly 110 bases away from here, from the GC box. So let's say our GC box starts at 100. Let's see, it starts at 1,112. So we want to be 110 downstream. Well, 110 downstream is 1,222. 1, 2, 2, 2. So we need to find 1, 2, 2, 2. That's going to be somewhere over here. No, keep going. A little too far. Let's see here. Looking down at the bottom there. 1, 2, 2. Look at that. 1, 2, 2, 2 is a C followed by an A. The most common combination of nucleotides for a transcription start site. So anywhere probably after this GC box, any CA, if this were human, and this is a turtle, and I'm not as familiar with turtles as I am with humans and flies, but if this were in a human, this is a perfect sequence for a GC box. This is the perfect length away for the transcription start site, and this is the exact right nucleotides for a transcription start site. So if you were to had this gene, and I asked you where was the transcription start site, your argument would be, well, I found the perfect consensus sequence for a GC box right here. And at exactly the perfect distance away from it, about 110 nucleotides, I found a CA, which is the perfect dinucleotide, the perfect combination of two letters for a transcription start site. At that point, this is your transcription start site. So yours may not be as easy to do as this one, but this is the kind of argument you should make. All of them should probably have something, should have a GC box of some sort in your promoter. It may not be as good as this one. It may only be as good as the green here, GGG, CGG, or maybe you have to reduce it to GG, CGG. But if you did, you should find one or more of these. Try to get as many of these G's in there as you can. Of course, if you get one with 4 on one side and 4 on the other, that's the perfect consensus. And if it's in the middle of your regulatory region here of your CPG island, that's it. So that's why I'm very confident that this is it, and I'm very confident that this is probably the primary transcription start site. Now, it's been proven that this may be the one where transcription start occurs most often, but it is not the only one that can occur, and probably any CA along here, perhaps a TA along here, could potentially start transcription. But this one is at the perfect distance from the GC box, so I suspect this is the one that's almost always used here in this gene. So this would be your transcription start site, and you would report that, and you would give the reasons for it. The reasons are, well, this position is exactly the right distance from a GC box, okay, 110. It has the right combinations of letters, a C followed by an A. And then, not only that, the GC box that we're using here is the perfect one. It's 4 G's, a C, and 4 more G's. And that's the consensus. That's the one that works the best experimentally. So since this is the best one, this is the perfect distance, and this is the perfect sequence here, this is you have a really good argument. Of course, the only real way to tell is to do the laboratory experiments and sequence the RNA and find out what the first letters are. And if the first letters of your RNA are a G followed by a U, then you know you got it right, because this would code an RNA for a G and a U. Wait, we are already in RNA here. This is the reverse. Yeah. So actually, if your RNA was a CA, that would be it. But this is how you would do this. This is probably your transcription start site right here at CA. Now, what if you didn't find this one? Well, if you had reduced it, maybe you could go to just GG, CGG. Let's say you searched and found none when you put GGG, CGG. So you reduce one so that I'm searching for GG, CGG. And that would have made it a little more difficult, because you would have found 3. You would have found the one we found here, but you would have found 2 others. And you would have gone through the same analysis, looked for a CA that's about 110 from here, and look for a CA over here. So here's a, let's see, Here's a CA. I don't know if that's 110 from this one, but if it were, if it were say 90, 130, somewhere in there, and that's the only one, that's your good argument. If you found one in here and it was 100 instead of 110, but it was the only CA in there, you could make that argument. So it's not perfect. You just have to make an argument for whatever it is you find. Now let's say you don't find one at all. You find no GC boxes. Well, you go on to the other promoters. There are others that are listed and you have to dig a little deeper in that in the worksheets. Find some other promoters that are common in CPG islands, and you search for those, and then do exactly as I did here. I have a feeling, though, all of yours are going to have GC boxes. Okay. Oh, before we go, I'll show you the other method that you don't have the ability to search for, but I do, and I wrote this program, so I know how it works. And apparently, it's the shape of the DNA based on the sequence, the long-range interactions, that allow something to be a promoter and to start transcription. And transcription start sites have certain properties that can be found when you look at the trend in a graph. And so I'm going to create a graph. Now I just happen to know I have to put in these numbers. Now this works with any CPG island. So I'm going to put in a window size, and I'll explain this in class when we meet after Thanksgiving. And then I'm going to put in, this one doesn't matter as much, I'm going to put in 24. And I'm going to grow this graph. Now this graph looks funny. I'm going to stretch this out. Hold on, so we get it stretched. I do like this because it allows you to stretch it. All right, now what we're looking for is we're looking for a place where the green line is peaking like this, and the blue and the orange are peaking together at a low point. So we want when this is highest and these are lowest. And if you go through this, all right, this doesn't happen very often, but it does happen right here. All right, right here where these are kind of dropping at the bottom pretty much at the same time this is at the top. And so where is this? Well, this is, the way you do this is it's going to tell you by over in the left-hand side here, I mean the right-hand side, you'll see that x equals 60.48, so 60.5. Well, we take that number and multiply by this number. So 60, what was it? 60, somewhere around 60. We'll just say it's 60. And we're going to multiply that by 24. So I'm going to go 60 times 24. Now you guys can't do this method, but I'll show you. 60 times 24, is that right? That's not right. Let's clear this thing. Alright, so 60. Oh, that was plus. Times 24. 1,440. So let's see what's at 1,440. Uh, 1,000. Where are we? 440. Like right about here, CA. Now, where is this? Well, this is a CA. It looks like a good spot. And where is it? 110 from this other one? Let's see. 1,000. So we want 1,330. 1,330. Let me find that. I'm there real close. 1,300 right here. So this is saying GCCGC. So it's not a GC box. But let's see, CTT. But I recognize this as another promoter. And what I'm going to tell you is you can have more than one promoter, but this method would tell me maybe that this one is something else, but actually I don't know the distance for that other promoter. But the other method, the scanning method, tells us that is a good place to look for a promoter. We would have preferred something probably about right here, over here, but this one is kind of peaking right here. I may have put the wrong numbers in. Let me try something a little different. Maybe I'm going to still put in 240, but I'm going to put in 32 on this one, and then we'll stretch it out. There are two optimal numbers to put in. And now here, it's clear that it's right about here. And this one is at, what is that about? Right about here. 45. We'll just say 45 times 32. Let's see where that comes out as. Is it the same place? 45 times 32. That's 1,440. It's about the same place. So this is pretty consistent. It's telling us that, at least by this method, that we would guess that the transcription start site is right here. All right. Now let's do something else. GGC, and no GC boxes up here. 1,320. Kind of close. There's some G's and A, but there's no GC box. That's kind of close. GG. But nope. So it may be that there is something other than a GC box here, a promoter causing this one to start. But you guys don't know that method. All right, you know this method, and actually this one just comes out great. As in, here is a consensus GC box, here's the consensus distance, and you find the consistent result. So you can't really go wrong using that method if you tell me that. The reality is that there's more than one transcription start site for a lot of these sequences, and depending on the method you use, you can find one or more of them. The only real way, of course, to find them is to do the wet lab work. Each of these different promoters, if you have multiple promoter sequences like data boxes and GC boxes, all of them will be able to attract the polymerase, and all of them will be able to start transcription. It just depends on what's probably going on in the cell. There are ways to mask these and cover these up. There are little RNAs that will stick to the DNA and cover some of these up. Depending on the pH, the other proteins binding in the area, they will cover these up. The histones might wrap around maybe this one so that it exposes this one, or maybe this one gets wrapped around the histone, depending on the methylation, right? You may methylate some of these blue boxes here, but not others, and it may cover up this one, but leave this one, maybe one of these others exposed. All of that can be a factor, and we just don't know exactly how this works yet, but the idea is we can begin to search for certain things, and this would give you clues as to what to look for, maybe in RNAs that maybe you're looking for RNAs that are related to this sequence, and you want to know, well, what do they start with? Maybe you're having a problem making primers or something, and you would like to know, well, am I inside this sequence or not, to make a primer to pull it out and do PCR. So at least some of this might give you some clues as to, well, should I look for primers, up here, down here, or maybe down here? Where should I look to make primers or do something if I'm going to try and actually I shouldn't say that RNA-seq doesn't quite work that way, but the idea is this method can help you find these start sites. And if you're trying to study how this is regulated, it might be interesting to know that, oh, it could start here, it could start here, it could start there, and all of those different places might alter, for instance, the splicing later on in the sequence. So if you start up here, you may splice a certain way. If you start down here, you splice a different way. If you splice down here. So all of these potential start sites might be related to the RNA that you're going to actually get after it's spliced and you have a mature RNA. Hopefully you guys have studied that. We're going to do a little bit more of that as well. The splicing is coming up, and I'll show you how to figure out where your splice sites are.